Dear students, in our series of basic courses, today we will continue the basic course of immunology, and our this lecture is about transplant immunology. So I hope you have seen our previous lectures. So how the story is started. So story of the histocompatibility that started with the transplant rejection and paved a way for the discovery of cell-mediated immune response by T cells has come to an end. We know that we have discussed already all these aspects in our previous lectures. But you all must be wondering that in spite of greater specificity of our immune system to differentiate between the self and non-self, how organ transplantation was made successful and which mechanisms are launched for the rejection of transplants in our body. And as we all commonly hear about organ transplants nowadays, so today's lecture is all about the pathways of foreign tissue recognition, its rejection, and immunosuppression of successful transplantation. So before starting the lecture, we must know that what is transplant and how many types it has. So if you look for here, so what is transplant? That is also called graft. So we have the two terminologies for that word. We called it as a transplant or we called also a graft. So what does mean by that? So that is a section of tissue or a complete organ that is removed from its original natural site and transferred to a new position in the same person or in a separate individual. That is called transplant. So that's what we should know before starting our today's lecture. So what does mean by the term transplants? So it's simply a section of tissue or complete tissue. So to remove from one's original site and transfer to the new position either in the same person or in another person. So that what we call it as a transplant. And then this transplant has four major types. As you can see here, autograft, isograft, allograft, and xenograft. So I hope maybe some of you heard about these terminologies, but even if you not heard about these terminologies, maybe being a biologist, we are familiar with these prefixes. So let's try to understand these terminologies by understanding their prefixes. So if you look for here, auto mean, we know that self, or graft mean again here transplant, but we have already learned. So auto is coming the same word as we call automatic, automated, or autoimmunity. So how we can define the autograft? It means a graft coming from the self and staying in the self, just like the word automatic, automated, or autoimmunity. So it is mean that is from self to self. Let's take the example for autograft, just like the coronary artery bypass, in which we take the leg vein to replace coronary vessel. So that is an example of autografts. Let's try to learn the second type of graft, that is called isografts. So again, look for the first word, that is the iso. Iso mean we know that the equal, and graft again mean transplant. So how we can simply define the isograft? So a graft coming from genetically equal person, just like the word isomer. Isomer, that mean equal formula. So here isograft is genetically equal person. So it means how we can say another terms, another way, from genetically identical person to self. In this case, if you took the example, It means from monozygotic twins transplantation. So that we know that if the two babies, they are twins, and more importantly, if they are a monozygotic twins, and we can retransplant the one organ from one kid to the another kids, if they are a monozygotic twins, so that is called isografting. And we see the third type of graft, that is called allograft, and that is actually our today's topic, which we are going to explain. Allograft, how we can define the word? Allo mean other, graft mean transplant. So how we can simply define this term allograft? So allograft mean a graft coming from the other person of same species. So it means from one human to another human. So that is called allograft. And in this one, how we can take the example, how we can learn the allograft, it's just like the word allosteric inhibitor. It means it inhibits the enzyme other than active site. So another terms, how we can also define, from genetically different person to self must be the same species. So that is example is, as you can see here, kidney transplant from a friend. So that is the example of allograft. And the last, which is mostly we use a term in case of different xenograft model for cancer and other diseases. So more importantly, in terms of cancer, we use this is a xenograft mice model and xenograft mouse model and so on. So what does mean by this term xenograft? So again, looks for here, we call it here, if you look at the prefix here, the prefix is xeno. And what does mean by this term? Xeno mean aliens or strangers. Or graft mean again the transplant, that we already know that. So how we can define? It means a graft that come from a person of different species, and just like from the different species to self. And in the example in this case, it means transplanting the pig heart valve into the human. It means, and similarly, if we talk about the mice xenograft model, so what we do in that case, what we do, we take the human cancer cells and we transplant into the mouse. So that's why that mice is called xenograft mice model or xenograft mouse model, if we are using this term. So how we can memorize these terms? So more importantly, know the prefixes, and you will never get these questions wrong. So you have to memorize the word auto, iso, allo, and geno. So of course, by remembering these prefixes, so we can memorize these types of transplants. In addition to these, I just want to share one thing here, which we are not going to discuss in detail, but that what we are talking about today's lecture, that is a rejection. Similarly to the transplant type, rejection also have a types, such as we call it as a hyperacute rejection, we call it the acute rejection, we call it the chronic rejection. Another type we call it as a graft versus host disease rejection. So that's why I'm emphasis here, what we are going to talk in our next slide. So if we know that, so what we are in the detail, we are going to the detail of allograft. So if you look for here, and we will elaborate with these types of, with the type of rejection types like hyperacute rejection, acute rejection, chronic rejection, and host and donor tissue rejection disease in that terms. So among all of these, most common transplant in terms of clinical application where rejection occur is the allograft. So in this terms, autograft, of course, no rejection, isograft, almost no rejection. This is something different. I'm not going to talk here in this lecture, but in today's lecture, mostly we will focus on the allograft. So that from where the name of pathway evolved in the recognition of non-self is derived. So there are, if you look for here, two pathways of allelo recognition pathway. So one is called direct allelo-antigen recognition, and other is called indirect allelo-antigen recognition. So if we go in further detail, before going to the further detail, once again, I want to memorize you about the types of rejection. So the first, especially, it involved in the hyperacute and acute rejection. And if you look for the next one, the indirect alloantigen recognition, that involved in the chronic rejection. So that you have to memorize in your mind. So let's try to understand what does mean by these pathway and how it happened. So if you look for here, the direct alloantigen recognition pathway. So what happening in this pathway? T cell recognize unprocessed allogenic MSC molecule on the graft APC, and recipient T cell directly recognize the foreign MSC molecule on the donor cell. So let's try to understand that phenomena with the help of this simple animation. So if you look for here, that is a recipient T cell and that we are called as a donor cell. So what is doing here in both cases, what you can see, this is a recipient T cell. So this recipient T cell, what is doing, it recognizing the MSC molecule of the graft APC. And then the recipient T cell directly recognizing the foreign MSC molecule on the donor cell. So you know, see, this is on the donor cell. So here you have donor MSC molecule and donor cell peptide, and recognizing, so it directly recipient T cell can recognize on the donor cell. So that's why they are called direct alloantigen recognition pathway. So what it does, so T cell activated by intact donor MSC protein. So that's T cell activated in intact donor MSC molecule directly. So that's why that is called direct alloantigen recognition pathway. And second, what we called it, the indirect. So what happened actually in the indirect? So let's try to make the understanding how indirect alloantigen recognition pathways works. So if you look for here, it happened due to the uptake and processing of alloantigen molecule by the recipient T cells. And when it is taken, of course, we know that then those T cells, what they do? Of course, when taken by the APC molecule, of course, how they do, we already understand all these phenomena. So what they will do with that antigen, they will process it. And after the processing, we know that they will convert that into their peptide, so antigen peptide. And now those peptides derived from the antigen molecule are presented to the recipient T cells. Let's again try to understand with the simple animation here. So if you look for here, we have a recipient T cells and we have a recipient APC, antigen processing cells. If you look for here, so now we have the antigen here. So antigen is already taken by the recipient APC molecule, then processed into the peptide, and now that peptide is recognized by the recipient T cells. So it means it's not a direct recognition, this is an indirect recognition. So if you look for here, this is a recipient MSC molecule and peptide is from the donor MSC molecule, but recognized by the recipient T cell. So this is an indirect way. So that's why that is called the indirect alloantigen recognition pathway. So what we are doing here, a T cell activated by the peptide from the donor MSC molecule. But in both cases, the APC molecule, they are from the recipient and the T cell are the recipient, but they are taken by these cells and presenting to the MSC molecule, and that is the, here we have peptide from the donor MSC molecule. So they are indirect way. So that is called indirect alloantigen recognition. So that's what we have learned in these terms. So now let's have a look exactly how this happens, how this all the processes, it means direct, indirect allele recognition, how it take place. So let's try to understand this phenomenon. So during, as you look for here in the slightly detail, so what we have here, actually this process take place in 3 steps. So first we call it as a sensitization. So what happened in the first stage, if you look for here, during the sensitization phase, the recipient T cells will show a foreign antigen on their surface. So I want to just clarify here. So this is a donor dendritic cells and we have a recipient APC. So try to understand this again. During the sensitization phase, recipient T cells, as you can see here, what it does, will show foreign antigen on their surface. As you can see here, they are showing a foreign antigen on their surface. And both recipient and donor APC will move toward the lymph nodes. As you can see here, it means these both are transport the alloantigens to the lymph nodes. So they are going to the lymph nodes, and when they're going to the lymph node, they're resulting in the immune activation. How is this immune become activated? If you look for here, and we already understand the phenomenon about that one. Of course, we have a cytotoxic T cells, we have a helper T cells, and we know that then they got differentiated into two special type of cells. One we call the effector cells, and other we call the memory cells. So it means they want to kill that foreign graft. So what they will change? They will change into the graft cells. So let's try to see what happened during the immune activation. So during the immune activation, there is the activation of T cells, and then these T cells resulting in the generation of effector T cells by direct or indirect pathways. So both the process is going to same way. Now what we have, as you can see here, okay, we have a number of effector T cells. So as we try to understand again from the sensitization. So what happening during the sensitization phase, recipient T cells will show foreign antigen on their surface, and both recipient and donor APC will move toward the lymph node, what we have learned so far, and where both of these will present the antigen to T cells, and both CD8 and CD4 positive T cells will be activated. And then as you can see here, we have both kind of T cells, cytotoxic as well as it means the CD8 and CD4 positive T cells. Now these effector T cells will move toward the allograft sites. So first what will happening? The migration of effector T cell to the site of allograft, where we have grafted that foreign section, whether it's an organ or whether it's a part of the tissue. And then what will happen at that the activation of effector T cells, or as a result, then the last step which is here, that is the rejection. So as a result, it will reject it. Why it rejected? Because these cytotoxic T cells, which is a CD8 positive T cells, directly kill the transplanted graft, while CD4 positive effector T cells help other cell of the immune system in rejection by secreting the cytokines. So how they're killing of the target cells by direct killing of CD8 cytotoxic T cells, and in other way the CD8 effector T cells will help the other cell of the immune system in rejection by secreting the cytokines. So they that tissue, that allograft part, or whether it's a tissue or whether it's a part of the tissue, it will be rejection by killing of the target cells and cytokine secretion. So this is the here what we have know that how the tissue got rejected. So it's a three-step process: sensitization, immune activation, and rejection. So now let's try to understand how the tissue could be made possible. So in order to understand how immunosuppressive drugs works, so what we have to go through, so first we understand what are the parameters for the possibility of the transmission we have to consider. So if you look for here, what we have to consider, of course, we look at the compatibility. So first, at least we should have the same blood groups. Whether the O positive should be transmitted the person who is donor and the recipient, both blood types should be the same. Whether if the recipient is O positive, the donor should be O positive. If the recipient is A negative, donor should be A negative. Why is so? This is a common phenomenon to avoid the blood agglutination. So that's what we have to need the compatibility, ABO blood group compatibility. This is one point. Another is the tissue typing. What we have to consider in these steps, we can do even by the testing that's called the HLA test. What we have to done, identification of compatible HLA antigen. That's the reason, if you remember, why isograft can't reject it, because we know that the twins have a same HLA antigens. But in that case, of course, we are getting from the other individual. Of course, they could not be same, but at least they should be compatible. So what other parameter we should consider before the transplantation? This tissue typing. And in what parameter we should consider here? Identification of compatible HLA antigens. Another important parameter, this is the use of immunosuppressive drugs. So how we can suppress the immune system? We can suppress the immune system by suppression of T cell activation. we know that the T cells got activated, and when the T cell got activated, they got attack on those cells directly or via secretion of cytokines. So both as a result of the rejection of that tissue or part of the tissue. So what we have to done, we should use certain kind of immunosuppressive drugs that can suppress of the T cell activation, or maybe they can lead to the T cell depletion. So that's what we have to understand here. So in that one, if you look for here, in order to understand how immunosuppressive drugs works, so flashback to our previous lectures on T cell activation. So if you want to understand this whole lecture, you can see our previous lecture about the T cell activation. So we must have to flash back to our previous lecture on the T cell activation. So what we have learned in that lecture, so we have learned about two signals for the T cell activation. So we know that the first is the release of IL-2. As we know that one we call a co-stimulation and one is a direct signal, signal 1 and that is the signal 2. This is the most important. Okay, we shall follow those two signals, okay, T cell activation and release the interleukin 2. So we know that that step is called the T cell signal, like B7 that is coming from the phagocytes, or we call that as the APC cells, and they activate the T cells. Another was the direct secretion of the cytokines. In that case, that was the IL-2, and that binding of IL-2 to ILR, interleukin 2 receptor, which give the ultimate signal for clonal expansion. As a result, these T cells start expansion of their same clone against that particular signals. What we know that process called the clonal expansion. It means, of course, that why clonal expansion? By the process of mitosis. So number of the T cells or T cell machinery start to be expand. So then those cells can kill those foreign tissue or part of the tissue. So let's, if you look for here, this is our previous lecture. So we will slightly go further. So let's elaborate this process to understand how the drug block the activation of these cells. So first takes a look here and try to understand slightly the detail how actually the tissues got rejected. So if you go for here, before talking about the drugs, let's zoom the T cells and try to understand actually what happened during the tissue rejection in those kind of inside the T cells. So because of the high diversity and specificity of HLA antigen, immunosuppressive drugs are used to suppress the T cell activation. So once a T cell is activated, a cascade of events started, resulting the release of, as you can see here, okay, we know that this is the antigen presenting cells, and we have a MSC molecule here, and they are recognized by the T cells when they got recognized. So what happened? When the T cell is activated, a cascade of events start, resulting in the release of calcium ion. So what happening after that? The calcium ion start to release. So you can see here, the first step is the release of calcium ion. So how the further process is going on. So this calcium ion bind to a protein that is called calmodulin, resulting in the release of the protein that is called calcineurin. So what the function is found by the calcineurin. So let's try to understand that phenomenon. That actually calcineurin is a phosphatase which bind to a particular factor that is called NFAT. So what it does actually and what it stands for, just try to understand. NFT is the stand for nuclear factor for activated T cell. So what happening? So when they got recognized, the T cells got activated, so result in the release of the calcium ion. Then these calcium ion, they start to bind with the calmodulin, and that calmodulin releases in the resulting in the release of calcineurin, and that calcineurin is a phosphatase, which phosphatase mean is added the phosphate group, and name is indicated that. So what it does the phosphorylation. It do the in this is a phosphatase, so it bind to the NFT and do dephosphorylate it. So as you know that this is phosphorylated, so what it does it actually, they start to dephosphorylate it. So NFC become dephosphorylated here. So this already was phosphorylated here. So now you can see that become dephosphorylated. So now what happens to this one, as you look for here, they move toward the nucleus, and as a result, what happening, they result in the release of the interleukin 2, and then these interleukin 2 bind to the interleukin 2 receptor, and then they activate another pathway that is called MTR pathway. And then that path actually involved in the process of mitosis in the cell division, and as you can see, a cell start dividing, and the number of T cells start forming, and number of the T cells is going to increase, and that what we call it as a clonal expansion. So this is how actually the T cells actually are working. So let's try to understand how we can block these pathways, and from where we can block these pathways if we want to make the transplantation successful. So what we have to consider here, so far in that one, so once a T cell activated, we have learned so far a cascade of events started resulting in the release of calcium ion, and then what happened, that calcium ion bind to a calmodulin, that result in the release of the calcineurin, and calcineurin, what it does, it dephosphorylates the NFT that translates toward the nucleus, then it releases the interleukin 2 that bind to the receptor of the interleukin and activate the MTR pathway, and that result in the cell cycle and result in the process of the expansion of T cells. So how we can stop that? By using a certain immunosuppressive drugs. So how immunosuppressive drugs work for the transplantation? So let's take the example here. Let's suppose we have a drug suppressing the T cell activation. In this case, let's suppose we have another drug, that is what we are calling here cyclosporin or tacrolimus, kind of the difficult name. Tacrolimus and cyclosporin actually inhibit this phosphatase. So if they inhibit this phosphatase, you can understand that what will happen, this NF will be not dephosphorylated. If it will not dephosphorylated, it will not move toward the nucleus, it will stay inside the cytoplasm. So it means the whole process, like the secretion of IL-2, then binding and MTR activation will stop. So there is no activation of T cell or no clonal expansion of the T cells. So what will happen? So is that inhibit the phosphatase. So NFAT, the nuclear factor of activated T cell, is not activated. Ultimately, interleukin 2 is not produced, resulting is in not proper activation of T cells. So this is one way how we can stop the activation of T cells. Another way, this is stopping here, as you can see here. So we also even have some other ways, like as you can see here, we have another drug that is called, what we call it as a belatacept. What it does actually, belatacept is an inhibitor of B7 and CD28 binding. So what we call, this is activation of the second signal. So how we call that? Because of co-stimulation. So what we can do that, we know that these B cell bind with the CD28, and as a result of this co-stimulation, the T cells got activated. So if we block this co-stimulation, so what will happen? The T cell will not got activated. So this is another way how we can stop, and we have the drugs to inhibit this process. The drug is called belatacept. So how it works? The belatacept is an inhibitor of B7 and CD28 binding. So it competitively bind to B7, ultimately rendering the T cells in energy. And energy means the T cells remain in an active form. They are not become activated. So we have another way here to block that, that is called IL-2 monoclonal antibodies. So what we call some monoclonal antibodies bind to interleukin 2 receptor, and that not let proliferate the T cells and formation of the effector T cells. So what will happen? Even this all process is going on, the T cells, but at the end, what we do, IL-2 is secreted, but we are not allowing to bind. So in this IL-2, they have their competitive antibody. So if they will bind with that receptor, so IL-2 could not bind. If this could not bind, MTR will not activate it and clonal expression will stop. So this is another way. So these are all the possible ways how we can stop the T cell activation. And by using these drugs, what we can do when we stop this process, what happened? And even what you can do that, another way, of course, you can do the drug specing the T cell directly activation like Th1, Th2, Th17 and T regulatory. So this is all the things by using this kind of particular drugs. So when we stop the T cell activation, so as a result, the transplantation become possible. So this is all what we have learned. So let's try to summarize all the data, what we have learned in today's lecture. So this is all is going on here. Even another important phenomenon here, even you can talk about, this is a sirolimus. What is this one? This is an inhibitor of MTR pathway. So even we can, at the last stage, even we can inhibit the MTR pathway. So if MTR pathway is inhibited, of course, then this whole process could be controlled. So the T cell again not got activated. So these are all the ways how we can stop the T cell activation. So let's try to summarize all the today's lecture in this slide. So what we have learned in the beginning, so we have defined the word transplant, that is also called the graft. How we define? We mentioned that a section of tissue or a complete organ that is removed from original natural site and transferred to the same person or another individual. That is called a transplant, and that is also called graft or grafting. If we take the few example like the hair transplant, we also discuss kidney transplant, heart transplant, and so on. So these are the different example of transplantation. So there are 4 types of transplants: autograft, we have already with the terms of their prefixes, allograft, isograft, and xenograft. And there are 2 pathways for alloantigen recognition. One is the direct, another is the indirect pathway. So tissue rejection result both because of the direct or indirect pathway. So direct recognition pathway is predominant in the short period after the transplantation because it's a direct pathway. So it gives a direct response, it's a quicker response. It means acute and hyperacute response is because of direct allele recognition. And but usually subsides with the depletion of messenger cells. If we talk about the indirect recognition, so while indirect recognition contribute to the continuous graft damage and play a role in chronic rejection. So if you remember, I have told you we have a 4 kind of rejection. What we call a hyperacute, acute, chronic, and host and donor disease competition. So what another type we have here. So transplantation is made possible by carefully matching the donor and recipient for compatibility. What we are compatibility? Like what is the ABO blood type? What about the tissue types like the HLA testing? We can know that, and then after that matching with these parameters, we can use a certain kind of immunosuppressive drugs. So this is a story how actually the tissue plant was rejected. That story was started with the MSc discovery, which we have discussed in our previous lecture. And in today's lecture, even we have discussed slightly more elaboration of the rejection, as well as we also discussed how the tissue transplant can become possible nowadays. So these are all the phenomena, and this is all about for today.